Today's speaker is Dr. Ravi Palanivelu. He's an associate professor of plant sciences here at the University of Arizona. Dr. Palanivelu received his PhD from the University of Georgia in genetics, and then he went on to do a postdoc at the University of Chicago. He came to the University of Arizona in 2006, and his lab currently focuses on understanding how plant cells communicate with one another, and he uses Arabidopsis thaliana as a model organism there. Dr. Palanivelu has over 30 publications in the field. He's mentored 16 plus undergraduates in his lab, of which several are UBRP students. And he's taught a variety of different classes at the U of A, which is how you may know him. He's taught different classes in biology and genetics, plant sciences, and I think he is now teaching PLS 360, which is Principles of Plant Physiology. And so without further ado, please go ahead and welcome Dr. Palanivelu as our UBRP seminar speaker.
Thank you, Jennifer, for the invitation and the kind introduction. It's very exciting to come and talk to all of you. As Jennifer mentioned, I am a big supporter of UBRP. I'm a big proponent. I advertise anywhere, everywhere. I talk to my colleagues and very proudly talk to them about this program because it's one of the preeminent biology research program in the country. This is not a cliche or this is actual truth. They started very early on and it's a wonderful program. And I want you guys to feel very good and proud to have joined this program because it is one of the few programs that has a great history and also a planned execution to get undergraduates a research experience. There's no doubt about it. And I myself had many fantastic undergrad students who were UBRP alumni who have gone on to do good things and they're doing good things. So I'm very pleased to come and talk to you guys this evening. So I'll keep it very simple, very general. This is summer, first week of summer. We all just unwinding from our semesters. I would love to talk about research, but I can always answer questions on it with more specifics if somebody has. So that's why I kept my title very general. It's a great time to be a biologist. Actually, it's always a great time to be a biologist, and especially now because of the explosion of genomics in every aspect of biology. So this is a great time to be a biologist. So I hope you all will continue to be in biology. So for today, I'll just not to toot my own horn. I just wanted to tell a little bit about myself and how did I get here, and also what do I do on research. So hopefully it gives a window into our life as professors. So I was originally born in India, where I did my. Okay. Good. I did my undergraduate in India. I was born in the southern part of hope you can see my cursor, in the southern part of India. Now I've lived more years of my life in the US than in India, but that's where my formative years were there. I did my undergraduate in agriculture, and I also did a master's in agriculture, although I spent most of my master's getting ready to come to the United States, but because that was my singular goal, is to come and do a PhD in the States. And so I made the trek from India all the way to Atlanta. My maiden flight was that, so it was very exciting. I started my PhD in 1991 at the University of Georgia in genetics. This building, actually, it was brand new when I joined. It was fantastic. I had a fantastic, great time. Within 2 semesters, I was very clear that I want to become a professor because I love teaching. Because as a grad student, we were asked to TA, and I really figured out that, I enjoy explaining stuff. So that was my foray into academia, and I wanted to stay there. As a result, after my PhD, I went to University of Chicago. I worked in this building, beautiful Ivy plants. So in this, because usually if you want to become a research faculty, one of the requirements is that they ask you to do postdoctoral fellowship where you get additional training, where you actually figure out what is that you want to have a career on and everything. So I early on went and started working on pollen during this fellowship. And that's where I figured out a project and then took it with me to come to University of Arizona in 2006. So a couple of things I learned, because I don't want to be too preachy, but just as a general scientist, I'm very lucky because I'm able to do what I wanted to do, so there is no doubt in my mind about it. But along the way, some lessons I learned, which I hope to share with you guys, is that you want to be very picky, meaning you have to have a clear plan of what you want to do so that you can go after it. But then once you pick, you have to be persistent, because most of the times you'll face failure, especially in research. 80% of the times there's a lot of failures. I don't want to say that as a discouraging thing. The way science is, because we do not know much about it, we figure out things and we may not be succeeding in, but that shouldn't discourage us. Instead, to figure out what is happening, because a lot of things are unknown. But persistence is very important. I'm sure that's true in any job. Whatever I'm saying is going to be true in anything. At least in academia, you have to publish, because publish is usually, publications are looked at as like, oh my God, they have to do all this. But in my view, it's actually one of the best things, because it shows to someone that you can take up a project, finish it, and communicate to others in a more structured and organized way that is well reviewed by other people, right? So it means a lot, because that means you have really figured out something and that has been accepted by your peers, all right? And always have a plan, right? I think that was one of the things that helped me along the way, is you want to know what you're doing so that you can equip yourself. Like, for example, you guys are doing this UBRP, right? That's actually a great thing, because if you ever go to a job after you finish your undergraduate, people will look at that and say, oh, they have done something besides their classes, right? So, and if you're already in a biology lab, you're research oriented, people will immediately, when I'm reviewing applications for grad school, that's what I'm looking for. It's like, oh, do you have any research experience? And it's really highly looked upon and everything. So always having a plan, and so that you can prepare yourself, is exactly what I would recommend, is that, so that the reason to have a plan is not to have a plan, but to help yourselves, because things don't happen overnight, right? Okay. And the other things that I also learned along the way is to have a good attitude. It shows, right? People they may not say to you, but they'll pick up right away. This person is going to be a great team player, great colleague, great friend, whatever, going to be trustworthy and everything. So what the attitude you bring to work is very important. And that served me well over the years. And appreciation, right? A lot of times, we're very quick to complain, but very slow to compliment. So always appreciate. Yes, so many things can go wrong, but so many things do go right too. So we'll just focus on that, appreciate it. So it gives a positive environment involved where we work. And I always sought out advice. I still seek advice because I don't pretend to know everything. Nobody should. And there's always, you can learn a lot from getting advice. But it's not enough if you just get advice. You have to absorb it and act. You don't have to follow on every one of them, but you can choose, figure out what works for your condition, for your situation, for your environment, whatever that is. And finally, I want to say, aspire and think big, because everybody can achieve and I've seen that over the years with my students. That's the biggest role as a mentor, is to believe in them before they believe in themselves, right? So for that, you should think big, and you can do it. Wow, that is the preaching section of my talk. It's over, and get it out of the way. But I hope I don't mean in a very bad way. I just wanted to share my life experience. And like I said, I feel very lucky. When I boarded the plane, I didn't have anything. Somebody gave me a fellowship in a university, and voila. Now both me and my wife are professors at the UFA. So we cannot be more grateful and feel lucky, right? But along the way, we did certain things right, we feel, and I wanted to share those simple things. Okay, so what do my lab work on? My lab, we are working on, so my lab is basically genetics, molecular biology, cell biology lab. We just happen to work with plants. But our fundamental questions are very molecular, cell biology, developmental biology. And so really specifically, we are interested in genes important for plant reproduction, right? How plants form seeds is what they're studying. I'll tell a little bit more about why that is. So I became interested in this because during my graduate career, I saw how cells interact with each other is very interesting to me in a multicellular organism. So here is one example, right? Especially during reproduction, there's ample opportunities for studying such interactions, right? So on the left side, you see a movie of a published work, not from our lab, from somebody else, mouse, sperm, and egg interaction. So if you look at this, it's really, you can watch it for hours together, right? It's really amazing how the sperm is so specific. They know go to the egg. Look at how everybody's trying, and one of them will succeed in breaking that zone outside and then enter, fertilize, and everything. That's mouse sperm, right? Almost only 5% difference between our genomes, between humans and mouse. Now, if you go to moss, very early on, right? Early plants, if you want to say, you can see that there's this sperm that's swimming and making these circles, but it knows exactly where it's going, and it will actually go to the archegonia, and it will fertilize, right? And this was the case with plants, that they were using motile sperms to achieve fertilization and reproduction. But if you come to flowering plants, which is what you see, if you just look out a window, what you see is a flowering plant, angiosperms, as they're called. They are the most dominant plants now in the world because they've almost colonized all parts of them, straight from the beach all the way to high mountain ranges. You will see an angiosperm, right? And if you look at that, let me see. Yeah. Okay. Now, in case of angiosperm reproduction, if you come to it, it's actually the sperm is not motile. It has lost its flagella in it, but still it has sperm cells. So they need help in delivering to the egg so that they can fertilize, right? And that's the job of the pollen. So inside every pollen grain that you ever see and ever encounter causes allergy. In fact, 80% of the people, if you just randomly stop them in the road and ask, what do you think of pollen? They'll all say allergy, right? We did a survey as a fun high school project. And so they always get the bad rap. But actually, we are in the way of pollen because they're trying to get to some other cell to deliver these sperm cells so that they can achieve fertilization. So every pollen grain that you ever encounter has 2 sperm cells inside, right? And if I remember, I told you that we were interested in cell-cell interactions, and I'm going to show you some movies to show how beautiful the system is, how you can actually use the system to study cell-cell interactions. So imagine a pollen lands on a stigma. This is Arabidopsis. Oops. This is Arabidopsis. You can see this pollen is football-shaped originally on the stigma. Then it germinates and sends out a tube, right? It becomes baseball shape. This goes from football to baseball shape. Within 15 minutes, right? Within 15 minutes of it landing on the stigma in Arabidopsis, it absorbs water and makes out a tube to go out, right? And you can see that. Let's watch it again. You can see that is actually absorbing water. It's swelling and it sends out a tube. I hope you can see the tube that went down. Watch how amazing it is, right? It is invading that cell without destroying it. So it's quietly, like a snake, passing it, because the stigma's job is to make sure the right pollen is given the opportunity to make a tube and go for it, right? So that's how all the angiosperms reproduce. They all have the pollen that land and then they start, right? So then what happens? So if you look at it, again, I'm just showing you 3 examples of these pollen tubes that have germinated. Their structure might be different, but they all do the same thing, which is they all grow down, the tube that grows down and try to go to the egg, which is sitting in these structures called ovules. We don't need to name the names, but they're deeply buried within the flower, right? And that happens in everything. So this is on the left side, I'm showing tomato, a very long style, or Arabidopsis, very short style, or in a grain like rice or barley in this example, where they have a very small thing. One grain that develops is actually, it has 2 stigma-like things, and then pollen will grow and germinate. They all do the same thing, pretty much the same thing. So the pollen tube is growing down, right? Where is it going? It's going towards, like I said, to the ovule. This is the close-up view of an ovule. This is a movie I made in my postdoc because I was very curious about like how is it attracting? How is it allowing things? And it was always difficult to study because they're deeply buried in the flower. So I developed a technique where we could scoop them out and put it on a petri dish and recapitulate everything that's happening in the wild, right? And I showed that actually you can reproduce everything. And since it is happening exposed, we can actually image them, we can study them, mutants, we can find the genes, and so on and so forth. So I'm just going to show you the final journey of the pollen tube, marched all the way. It's coming to this structure, and this is the ovule, and inside it, the egg is sitting here, deeply buried. But there are a lot of helper cells that are bringing this pollen correctly into the thing. Right? If you watch it, we tag the pollen with the GFP marker, and it correctly goes, and then it explodes. Right? If you see the end, it knows that is where the egg is sitting, and right there it goes and explodes. And in that explosion, it slings the two sperm cells, and then one sperm cell will fuse with the egg, and the other will fuse with another gamete, and then this becomes the seed, the future seed. And this is how actually all plants reproduce. All the flowering plants will reproduce. And that's why in flowering plants, you have something called double fertilization. You have two fertilization events, two sperm, two female gametes, and they fuse, and you need both to get the seed. If you had only the sperm and the egg, you get an embryo. But that embryo needs to be nourished. And that's what the second fertilization product does, is to give that thing. But that actually brings me up to the main point, is that it's also commercially important. Right? For the plant, it's absolute. This is the way it reproduces. Why is that? That second fertilization product, I said, is what develops into what is called as endosperm. And that's what we eat. So it's not an overstatement if I say 80% of the human food that we eat comes from endosperm, right? In the afternoon lunch, if you ate a wheat bread or a rice or a corn, what you ate was endosperm. The endosperm is the one that has the nutrition. So in a peanut, it's stored as the endosperm stores the energy as oil and nourishes the embryo. In a bean, it stores it as proteins and nourishes the embryo. We eat that. In a grain, cereals, it stores carbohydrates and in the endosperm, and then we eat that. But literally everything we consume as food is from that. So you might think like, oh, what about animals? We feed them and then we eat the animals. But regardless, it's not an overstatement if I say, if angiosperms are wiped out, you'll not have civilization. It's very difficult, right? Yeah. And also like spices, you name it. Every time you see a seed, you should think back and say, that came with the help of a pollen grain. That's what helped to fertilize and gave that seed. So it's important for the plant. We are also using it for food. So here I'm showing an example with the tomato life cycle, flower and produces. That fertilization is what makes the seed. Sometimes we don't eat the seeds. Like in tomato, we don't eat the seeds, but we eat the fruit. That fruit is stimulated because that fertilization happens, right? So either we're going to eat the fruits or we're going to eat the seeds. No matter what, our food and nutrition is really dependent on flowering plant reproduction, right? So I'm just showing you on the bottom all kinds of seeds we eat. Right? In fact, we do an outreach thing for high school students and middle school, like no pollen, no seed. Right? We teach them how it's very important. Okay, so now, so make sure, okay, I have 5 minutes, I'll finish it up. So what we are doing is that we are studying how this pollen grain, with nobody's intervention and help, can start its journey and make it all the way and successfully fertilize. It's a long journey. In corn, it takes 2 days. You see the silk, right? That's hanging from the thing. That's each one is supporting one pollen grain to grow. And then it fertilizes, you get one corn kernel, right? Then when you have many, you have the cob. Like that, you just think about it. Like what is also amazing is that there's hundreds of pollen floating around, right? How do they discriminate each other? How do they find their partner? How does it, a stigma many different types of pollen can land on, right? So starting from that point, and then how does it make the tube? How does it know, okay, I have to turn this way. I have to slow down here. I have to stop. I have to burst. And all of that step is actually highly coordinated by interaction between the cells that direct the pollen tube to get to the point. And so I love this system, and I thought this is a great system. We do not know much about it. It's fundamental, right? It's also it's the crux of evolutionary biology as well. This is where speciation happens. If they're two different species, they won't support the pollen tube growth, and they can't fertilize. Then hybrids cannot be formed. So they isolate themselves further, right? So speciation is driven by this process. So I really love this idea that the system is at the crux of everything, right? It's very important, commercially important for the plant. We do not know much. There's a lot of cell-cell interaction. In fact, pollen is the only plant cell that will have to interact with 7 different cells before it reaches the germ. No other plant cell will have to go through that criteria. Right? You'll always have cell-cell interaction between higher eukaryotes, right? Take a root or a leaf. They're all coordinating with each other. But there's only coordination and talk with neighbors. But pollen is the only one that will have to grow past 7 different cells before it reaches the target. And then, so that fascinated me. Like, how does it know? It's like a relay, right? Every time it interacts with a new cell, it has to reset its program, getting ready for the next one, and then it has to correctly deliver the sperm. It's carrying a very high-priced cargo to do. So this fascinated me, and that's what my lab has done over the last 15 years, is identified the genes that are important along the way. So then you may say, like, where is this all going? This is all basic research. So what? Who cares? So great, you're interested, but it's up to you. But I think this is where I want to stress that these are the discoveries that are important, that will help us in the future. I'll give you one example. If you ever want to break a species barrier, you got to know how compatible pollination works, so that when they isolate themselves, you now know the lock and key. Then you can match them and break the species barrier, right? So we finding the genes are helping to break species barrier. We already, in the last couple of years, we have found a protein that's very important in overcoming hybridization barriers. And we are able to break the barrier. We can now cross 2 different things earlier, which we could not cross. We now put the protein from plant 1 into plant 2, and now plant 1 can accept the plant 2 pollen, and it will fertilize. So it will help us to break the species barrier. Or the flip side is, if you understand this, we can also create a species barrier. So there is a lot of interest in people saying that, if you make a transgenic pollen, you may spread into the native species and everything. By using the tools that we develop, one can engineer a plant with it so that nobody else can fertilize. Right? For example, I'll give you a simple example, right? When you go to a hotel, when you check in, they give you a card. They don't come with you to make sure you go into the right room. They know that with that key, you can only enter one room, right? Similarly, we are thinking of coming up with a tag on the pollen and the female so that only they can match, and then only fertilization can happen. So in other words, we can create a barrier wherever we want, so that you can then release these varieties without worrying about them crossing with native plants. Right? And of late, by the way, that is actually a major bottleneck, right? Regulatory agencies want these things to be solved before they can release to farmers or consumers and industry. So this is a big regulatory block is there, and we feel that we can contribute. And of late, we have actually, I know Jennifer introduced us with the Arabidopsis. We are now moving into tomato, into a crop plant, because of the primary knowledge we gained from our studying these systems. We now know that the pollen tube growth and fertilization is highly vulnerable to heat stress. With the warming climate, we are going to face problems, because we found that even raising 1 or 2 degrees, fertilization is affected. So many times, like people will ask me when I go to these outreach events, they'll say, planet is only warming by 1 degree. It's not a big deal. What's the thing? So I tell them, a simple example is that, so your body temperature is 98.4, right? What is fever? Just 1 more degree, you have a fever. You come almost paralyzed, right? So 1 degree is a huge change, because our biological systems have adapted to certain functioning under certain conditions. So rapidly increasing the temperature is not a good idea, because they're not going to survive. So we found that reproduction is very vulnerable to heat stress. And if you have any problem in that, you can actually wipe out the seed and fruit formation, which is, I told you earlier, that's the basis for all our consumption, right? So we found that heat susceptible Heinz, which all the cultivated tomato varieties are heat susceptible, right? So even during pollination, if you get the heat stress, all other times the plant was normal, right? Before or just during that phase, if you give heat stress, it's finished, because we saw that nearly half of the fruit yield went down. Whereas heat tolerant varieties, we found that they only reduced by 16%. So we think that there is a genetic basis for this that's conferring this tolerance to these tomato varieties. And we are now started a new project in the last 1 year. So I'm looking for students actually. So we could support. And we are now finding that the idea is that if we find these genes that help the tolerant varieties to successfully set seeds and fruits during heat stress, we can help with, we can do precision breeding and transform the cultivated tomato varieties, which are heat susceptible, into tolerant ones. And we also believe that this technology can be used in all other crops, where we could figure out many of the crops we consume are heat susceptible. So there is a big application in this prime base. What appears as a basic thing is now coming to fruition, no pun intended, in order to help us in generating. So with that, I'll stop and thank my graduate advisor, Richard Meager, and postdoc advisor, Daphne Bruce, who gave me an opportunity to mentor and help me develop. And I also want to thank my current and past members of my lab who have done all the hard work, who are phenomenal people to work with. It's really fun, not just good science, but also fun. And also the funding. None of this will happen without funding, So during Corona times, like people always, have a public doesn't understand that basic research is what helped us. I don't know how many of you know the RNA vaccine was nothing, right? Nobody even knew about it. But in the 90s, people were already, researchers in Penn State, University of Pennsylvania, were developing an alternate mode of vaccine. And that's how mRNA vaccines were. It was not discovered just last year. The idea of delivering vaccines by mRNA was a basic research in a research lab with seemingly no outlet or an application. But it was developed. It was scorned in the early days, but she kept at it, who went on to find the Moderna company, right? And that was simply supported by NIH, National Institute of Health. So that is, only government can take that risk and support curiosity and fund them. And eventually it comes back to help us as a society. So basic research is, in other words, everybody wants application, but you can't apply without basic research, right? And that starts with curiosity of people like us, and the future, like with you guys, to keep this line moving so that our society will benefit. But I'm grateful that government gave us funds to keep studying what they're doing, including UBRP. Many of my students have been wonderfully supported by UBRP, who have gone on to grad school now or to companies to work.